So now, does this mean we always move left to right? The answer is no. We always read 3 to 5 and build 5 to 3. So genes, the top strand can sometimes be the template strand. The bottom strand can sometimes be the template strand. It really is determined by where the promoter lies. So I'm going to use a term here. I'm going to try to define it. Upstream. Upstream means, in molecular biology terms, before. So the promoter here is upstream of the gene on the left-hand side. So if it binds here, this becomes the template because I always read 3 to 5. Here the promoter, again, upstream here on the left-hand side, making, again, the bottom strand the template strand. But imagine a scenario now where the promoter is upstream of the gene, but now is on this side, the right side. Well, now remember, I read 3 to 5. So now this becomes the template. And that's sort of important because now either strand can serve as a template strand. And so there may be times when there's a promoter, a weak promoter here as well. And so if the promoter's here, the other strand becomes a template strand, meaning now that we can pack a lot more information into our DNA if either strand can serve as a template. So here we have the corresponding RNA here, running 5 to 3 in this direction. Here we have the corresponding RNA running 5 to 3 in the opposite direction. So the promoter is extremely important in orienting RNA polymerase and determining which strand is the template strand.
In bacterial cells, the transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA molecule, leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site.
So let me just quickly just try to explain what they're trying to do here, right? So what they're doing is they're setting up that coding region by showing where the terminator site is in the promoter sequence. Now, what does -10, -35 mean? Well, remember that we talked about the fact that we can have upstream and downstream positions. So we know that the promoter is upstream of where the gene starts. Well, we're going to call the start site. I apologize, I don't have much white space here. The start site of a gene is called the +1 site. That's where transcription begins. That's where we start to copy. Anything that comes before that is now in the negative side of the ledger. So what this means is that 10 bases upstream of the +1 are sequences. 35 bases upstream of the start site are other sequences, of which now make up the promoter. So all the sequences, 10 bases to 35 bases in front of the start site, represent the promoter sequence.
The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule.
So here's essentially the 3 steps of initiation. RNA polymerase, using the sigma factor, is going to bind to the promoter DNA to form a complex called RPC, meaning closed complex. It hasn't unwound the strands yet. And there's an equilibrium there, right? Because there's a binding constant. RNA polymerase is going to recognize and bind the promoter with a certain specificity. Well, just as it binds, like we saw in Chapter 9, just as it binds, it also can fall off. So there's an interplay here. Now, of course, the better the RNA polymerase binds a promoter, the more it favors closed complex. But listen, that's concentration dependent. Now, what we're going to see is the committed step, which I keep bringing up, is when the closed complex transitions to an open complex. Notice there's only one arrow here. There's no equilibrium. Once we open it, it's so energetically unfavorable to open it, we might as well finish it. And that's what we're going to see here in the next step.
By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up.
So as the RNA polymerase travels, so does the bubble. So does the open complex, continuously exposing new strands to use as template so we can build the growing RNA.
As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding of the Rho protein to the RNA.
So listen, Rho protein, have a basic idea what it does. We're not going to get much into the mechanism of it, but I at least want you to appreciate and watch how we can regulate transcription through a series of regulatory proteins like Rho.
It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase. Between the Rho recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription, resulting in the full-length mRNA transcribed from the gene.
Here's what I love about that, and maybe you don't appreciate it because it's somewhat subtle, but the single-stranded nature of RNA gives us so many opportunities to do interesting things. You can see there, while it's single-stranded, it does have the ability to fold onto itself. It formed what we call this hairpin loop here, and that's changing, right? The alteration of the RNA conformation actually caused the process to halt and allowed the Rho protein to carry out its stated function, which was to separate the transcript from the rest of the complex. And so we often use secondary structure within RNA as a way to regulate a variety of processes in the cell. This was just one example.